So, I want to talk to you about the Unit 5 test. This is your first major test of this semester over chapters 13, 14, and 15. Most of the test is going to come from chapters 13 and 14, meiosis, and I guess I would call it more basic genetics. Chapter 15 really dives into some of the complexities of genetics and goes into very unique situations. Part of that is the sex-linked traits, and there will be plenty of questions that you have on sex-linked traits on this test, and we'll go through all the questions on the test here in a second. But outside of that, there is a lot of vocabulary and concepts in chapter 15 that won't come up on the test. And to help give you an idea as to how to focus, I'm going to talk to you about the test right now. There are 50 multiple choice questions, no written questions. Okay, so there's no essay questions on this one. This is kind of how we did, I think, one of the tests last semester. Now, I say there's no written questions, but on quite a few questions, you will probably need to write out a Punnett square and fill it out. There should be room on your test, like a blank back or the cover page. You might also want to just have a scratch sheet of paper with nothing written on it that you use to do some of the practice problems. So to start out with, the first question I have is about sexual reproduction versus asexual reproduction. So know the difference. The next few questions, about the next 4 questions or so, deal with chromosomes and chromosome number. Now, I think all of you know a normal diploid number for humans is what? It's 46. That's right. That's our diploid number. Meiosis, remember, is going to do what to that number? It's going to cut it in half. You actually get 4 cells, each with 23 chromosomes. When you have a diploid human cell with 46 go through meiosis, you end up with 4 cells with 23 chromosomes each. Now, I've talked a little bit about this, and your book did as well, and you'll see this notation on the test. But when we talk about diploid, we are saying 2 of each chromosome. So you'll have a certain number of unique chromosomes. When we use the term diploid, we have a pair of each of those. We have 2 of each of those unique chromosomes. And there's a notation I've been using, or that your book's been using. I haven't really used it a whole lot, but I will on the test. 2n, like a number 2 and then a lowercase n. That is the symbol for diploid. Again, your reading's gone into that. I've mentioned it a little, but we haven't really spent time on chromosome number in a while. So if you see, like in humans, 2n equals 46. That means the diploid is 46. And so what symbol do we use for haploid? Just the letter N, which is an understood 1n. So if you see 2n equals, or just the letter n, lowercase n equals, the 2n means diploid, the n, 1n means haploid. So there will be a couple questions about, if this is the diploid number, what would the haploid number be? Well, it would be half. What might explain, say, an extra chromosome? How could that happen? Things of that nature. I ask a bit about homologous chromosomes, which are simply what? What does the term homologous chromosome mean? A homologous chromosome is, you got a chromosome, its homologous chromosome is the matching pair. Now, it might not be the duplicate. You do get sister chromatids, which are copies of those. But when we use the term homologous chromosome, we specifically are talking about like chromosome 1 will have a matching chromosome 1. Those two are homologous. And it's kind of like the lab we did, the lab activity. You were trying to find the homologous or matching chromosome for each one. It's a great example. Anytime there is a gene for, say, eye color, skin color, flower color, whatever the organism or the trait may be, anytime there's a gene for a trait at that same location on one chromosome, there is a gene for that trait on its homologous partner. They may not be the same gene. It might be a brown-eyed gene and a blue-eyed gene, meaning different alleles, but it's a gene for the same trait at that same position on matching chromosomes. What are the human sex chromosomes? X and Y. Know what makes a female, what makes a male. The only reason you have meiosis in most animals is to make sperm and egg cells, to make haploid cells known as gametes, sex cells. So I'm noticing here there is one question where I give kind of a diagram of diploid to haploid and where meiosis comes into play, where fertilization comes into play. Understand the general cycle of sexual reproduction. Diploid individuals, through meiosis, make haploid cells. Those haploid cells are the sperm and egg cells. They fuse together in fertilization to make a zygote. And through mitosis that we discussed last semester, that single cell that all of you start as will grow into the billions and billions of cells that make up an adult. So understand that general cycle. Questions about 10 through 20. There's about 10 questions where I give you a word bank of prophase 1, metaphase 1, anaphase 1, telophase 1, prophase 2, metaphase 2, anaphase 2, telophase 2. Those 8 distinct phases of meiosis. You'll have a word bank, and then I will have about 10 events where I talk about crossing over or spindle fibers forming under certain conditions or chromosomes lining up or homologous pairs separating versus sister chromatids separating. Those kinds of events you will have to basically match to one of the phases of meiosis. So matching events to phases of meiosis. We had questions just like that in the mitosis test, but we'll be doing it for meiosis this time. So that takes us through about question 20. There's one where I show you a karyotype and you just tell me about the individual, maybe gender or if there's an abnormality or not. I ask a little bit about crossing over, where
In that question or
sorry. As a separate question, I would ask about crossing over, yeah. So there's a karyotype question, then another question where I ask about crossing over, which remember is just when pieces of homologous chromosomes will switch places. So you kind of shuffle what alleles that chromosome might have by trading them with the allele for that same trait with another chromosome. I will ask, and these aren't necessarily together, but there are 2 genetics laws. One is called the law of segregation. The other is called the law of independent assortment. I would go back to your book and read about them. We mentioned them. The concepts that those laws dictate are things that we kind of have naturally done when we filled out Punnett squares, but I would certainly review those two laws. Those are two what we call Mendelian laws. So, that relates back to Mendel and some of the original experiments in genetics. I'm noticing a question where I describe a trait and the genetics behind that trait, and you're going to tell me whether or not it is codominance or incomplete dominance. So know the difference between codominance and incomplete dominance. And that takes us about halfway through the test. And we haven't done a single genetics problem yet. I get into a little bit of discussion. There are a few questions about blood types. So understand that there's 3 unique alleles. There are A and B alleles for type A and type B blood that are actually codominant to each other. They will both show up in people with type AB blood if a person has both those genes. And then the third allele is a recessive O. So O is recessive to A and to B, but A and B are codominant to each other. Again, understanding the concepts behind that. Along those lines, I would know the difference between autosomal genes and sex-linked genes. Those are 2 very important terms to distinguish, autosomal and sex-linked. In the case of humans, out of your 23 unique chromosomes, you have 23 pairs of chromosomes in most of your somatic cells. 22 are what we would call autosomal chromosomes or autosomes. The last pair are your sex chromosomes. So when a gene is on the X or Y chromosome, if a gene is just on one of those chromosomes, that's when we call it a sex-linked trait. And for girls, we have to write out XX, and for boys, we write out XY with superscripted letters for the alleles. If the gene is on one of the 22 pairs that are not determining of your gender, then you just write it normally like big A, or big A, little a. That's how you would write autosomal. But you do write them differently, and that affects your Punnett square and how you read it. But I will tell you if a gene is autosomal or recessive, or you can tell whether it is based on how I write the genotypes. But in a little bit, I'm going to talk about pedigrees, where we can use pedigrees to discern whether it's autosomal or sex-linked. Okay, so basically between blood type and, autosomal versus sex-linked and some of the genetics laws of segregation, independent assortment, we're to question 30. And on question 30, and again, this might change a little. I might, as I reread this test today, decide I don't like that question, and I might just delete it and add one to the end. So when we get the official final version that you take on Friday, maybe this will be question 29 or question 31. But around question 30, you'll start dealing with crosses and Punnett squares. So I'm looking at 1, 2, 3, 4, 5, 6, 7, 8. About 8 questions in the 30s here are questions where I tell you, like, this trait is recessive, this trait is dominant, these are the parents, what, ratio or percentage of the offspring would look a certain way. A few of them are super easy. They might even be as simple as, short pea plants are recessive to tall pea plants, and you have 2 short parents, meaning they're both homozygous recessive. and you do the Punnett square, and every kid is homozygous recessive. Both parents are short, all the kids are short. The answer might be 100% or 0%, depending on the question. They may be very straightforward and simple like that.
Are we gonna have to know how to do the ratios and stuff, or like are we always gonna be able to
Well,
do percentages? Because I'm better at percentages.
I typically, the percentages are typically used for the 2x2 Punnett squares. So it's 0, 25, 50, 75, or 100%, depending on whether it's 0 through 4. When we do the 4x4s, instead of saying 12.5%, I will say like 2 out of 16. The only time I use percentages are when they are 25, 50, 75, or 0 and 100. Very obvious percentages, something out of 4. Once we get to the 4x4 Punnett squares, I will say like 8 out of 16 instead of saying 50%, because sometimes out of 16, the percentage doesn't really work out. I might ask about ratios in a like 2 to 2 or 1 to 2 to 1 ratio. So, I mean, that's basically the same thing where I'm saying it's out of 4. Some of the simple 2x2 Punnett squares will be like blood type related. We had a couple on that blood type sheet where I asked like who could be the father, who could not be the father. So you might have a situation where there's a child with type O blood, or I tell you there's a child with type O blood, what blood types could the father not have? And basically the answer would be he could not be AB, because that would mean he did not have an O to give to his type O child. Look through some of those like parents and child, blood type, who the father could be, who the father couldn't be, kinds of questions. So be familiar with blood type. I actually seem to hit it on a couple questions. After that, I do have one problem at least with sex-linked traits, where say it's on the X chromosome, and then I ask about sons and daughters and what gender might be affected, or you'll have to do a 2x2 Punnett square, but the mother will be XX, the father XY, and then you'll have to sort out the children that way. So look at those review problems. And that takes us up to about question 40. In the 40s, out of the 50 questions, I have one pedigree with 2 questions. And usually the 2 questions are, is the shaded trait dominant or recessive? And is the shaded trait sex-linked or autosomal? Meaning, is it on the X chromosome or not, basically. So be familiar with those, and I can online or in class right now or at office hours throw some examples up. So make use of the time between now and then if you want some practice on that. There are 3 questions that I'm looking at here where I give 2 autosomal traits where you do not write XX or XY, but there's 2 traits, so it might be like big T, big A, little a, cross with big T, little a. And you'll have to do a 4x4 Punnett square and tell me the proportion of offspring that, say, have one trait with another trait, or
Like that worksheet that we did.
yeah, the one from just the last class, which on the coursework page is posted for today. I have the 2, those 2 problems on the most recent worksheet in the Keys Online, that actually had a sex linked with a autosomal trait. So you were considering both at the same time, but it's still a 4x4 Punnett square. These will be kind of the standard 4x4 Punnett square, which I had the dihybrid practice problems. We did have a whole sheet of 4x4 problems that you'll want to review. The last problem that you have to do a Punnett square for is like the 2 practice problems we did kind of last class, and that's posted for today, And in that problem on the practice sheet, I had hemophilia, which is a sex-linked trait, being considered simultaneously with an autosomal trait of blood type. So we looked at hemophilia and blood type, and we got these weird genotypes that were like X big H, little o, crossed with X something Y, A, B, or so, along those lines. They look weird when you do a sex-linked trait and an autosomal trait together in a dihybrid 4x4 cross, but they work the same way. So don't ever be thrown off by the weirdness of the symbols. And then, oddly, I just decided, oh, well, this is kind of a focus on chapter 15. I have a couple questions where I ask about monosomy and trisomy, where you might have one of a chromosome instead of two, or you might have three of a certain chromosome instead of two, basically meaning you're missing or have an extra chromosome. Again, the activity we did last class, you may or may not have had a karyotype that was abnormal.
3 of a chromosome is Down syndrome, right?
Now, if it's trisomy 21 specifically, if the 21st chromosome is the chromosome that has 3 chromosomes instead of 2, that specifically is Down syndrome. But you can get other chromosomes. Like if you have 3 chromosome, I think it's chromosome 13, that is a different syndrome. I believe its name is Patau's syndrome. And it's a disorder because of the abnormal number of chromosomes, an extra chromosome 13, but it's not called Down syndrome. So Down syndrome is an extra chromosome, but only if it's chromosome 21. You do not have to know any syndrome names. I would, in the question, explain or show you a karyotype. I could just show you karyotype, and you can look and say, hey, there's 3 chromosome 15s there. That is trisomy 15. Or you might look at a karyotype and say, hey, there's only 1 chromosome 20. That's called monosomy 20, where you have 1 of that specific chromosome. So finally, I want to ask you real quick, this will be a fun one that relates to one of the final problems. What is the name that we give to the event where the wrong number of chromosomes goes to one side? Like instead of the sister chromatids separating in anaphase 2, maybe they don't separate and they both go to one gamete together, causing a gamete to have an extra chromosome. What do we call it when the chromosomes are not pulled apart properly by the spindle fibers? Anybody remember that term? I mentioned it briefly at the end of our lecture. That is called non-disjunction. You should know that term and know about it. So if you are not familiar with that term, know about it. So that's kind of where the last couple questions on the test end up, is with some of the chromosomal disorders of information from chapter 15. Again, 50 multiple choice questions, no written essays. Because you're doing a couple of Punnett squares, depending on how efficiently you do them and if you do them correctly or not, you may find that the 70 minutes you have for the test is plenty of time, or you might be struggling to finish the test on time. So once the test starts, I would try to work somewhat diligently and, maybe star a question and come back to it if you're not sure, but make sure you've gone through the whole test. We have office hours tomorrow. Unless your second period, you have office hours on Friday before the test. I should be in my room by 7:30 on Friday if you get here early. I can be online pretty early on Friday for questions if you want to use it that way. And that's it. Good luck on the test. Let me know if you have questions.